Doctors Beggs and Parsons discuss the current status of gene therapies in rare neuromuscular disorders in this 8-part podcast series. This is derived from the symposium that was presented at the MDA 2025 conference in Dallas, Texas in March 2025 and is intended for healthcare professionals only. This podcast includes information about investigational compounds that do not yet have a regulatory approval or authorization for a specific indication. The safety and efficacy of the agents under investigation have not been established, and contents of this podcast shall not be used in any manner to directly or indirectly promote or sell the product for unapproved uses. The ASPIRO clinical trial is on clinical hold since September 2021.
The views, thoughts, and opinions expressed in this presentation belong solely to the author and are subject to change without notice. The contents of this presentation do not constitute an endorsement of any product or indication by Astellas.
In this part, Dr. Beggs will provide an explanation of AAV-mediated gene therapies.
AAV vectors, which I'm going to be talking about more today, or adeno-associated viral vectors, or small viruses. Their DNA gets delivered into the cell and remains extrachromosomal. There are very rare occasional integrations, but the risk of oncogenesis as a result is significantly lower. As a consequence of remaining extrachromosomal, though, we do have to think about what happens as the cells divide, and potentially the durability of treatment is more limited. So there have been a lot of movement and development over the years, starting back in the 1980s when the first AAV genomes were isolated and sequenced. This led to development of methods to produce recombinant AAVs that would lack the genes necessary for viral replication, but contain a therapeutic gene you wish to deliver. Through this, the structure of AAVs have been developed. There have been isolation of a number of naturally occurring variants. So you've heard of AAV8, AAV9, also RH74, derived from a rhesus monkey for the RH. These have all been used in clinical trials. And then at the end, I'll talk a little bit about directed evolution methods to actually engineer capsids with particular properties that are beneficial. So throughout this, we've identified some of the issues that arise in this. It was initially thought that AAV vectors were non-immunogenic, but in fact there are immune responses, not just to the viral payload, to the therapeutic protein, but also to the viral vectors, and you're going to hear about that from Dr. Parsons. Over time, as we've come to understand these challenges, we've also been developing approaches to mitigate them. So in terms of clinical trials and treatments, the very first studies were done back in the 1970s. But by the early 2000s, the very first clinical therapeutic was approved in China. It was actually an oncolytic virus carrying a P53 gene to treat head and neck cancers. And by now, there are over 40 approved treatments for various types of AAV-delivered gene therapies. And of course, the ones we know a lot about are Zolgensma, which was approved in 2019, and Elevidys, which was approved last year. So number of challenges, and then also a number of approaches to overcome those challenges. So first of all, the preclinical data are not always sufficient to predict the response of a human patient. So for example, in X-linked myotubular myopathy, we had mouse and dog models that exhibited a myopathy, but nothing else. And yet when we treated human patients, we discovered that patients with X-linked myotubular myopathy actually had a previously only poorly recognized hepatopathology that led to potential liver consequences following gene therapy. So the animal models don't always predict the clinical outcome in humans. Also, we have small disease populations. These are rare diseases, so it's important to understand the natural history of these diseases, understand the heterogeneity among the clinical population. And it's very important to engage with families and with patients and communities, understand who might be at increased risk to treatment with one of these. This feeds into safety considerations. We need to think also about some of the immune responses. I think we're starting to learn, for example, with the gene therapies for Duchenne, we know this from SMA, that some patients get into trouble and others don't. We need to understand why that may be. And we don't know about the long-term effects. This has been very recent. So are there long-term risks? And what is the durability over time? And then finally, regulatory guidance is evolving, both the EMA and the FDA. This is both a challenge and a benefit, I think. As regulators come up with appropriate ways to do things, I think it makes clinical trial design a little bit more straightforward and benefits everybody through the use of best practices.
In the next part, Dr. Beggs will discuss mitigation strategies to address challenges in the development of gene therapy programs.